Okay, this is the Honors Bio Unit 8 Study Guide slideshow. So the top is about a karyotype. It's an image of one's chromosomes. Use this karyotype to answer the following questions. Identify the gender of this individual. Well, this individual is a male, okay, because you can see X and Y. How are chromosomes organized? By homologous pairs. See, they're paired together here. Same size, and in the other slideshow that you did, it was by same banding pattern. Which chromosomes are autosomes? Pairs 1 through 22. How many total chromosomes are shown? 46, 23 pairs. Which type of cells are these chromosomes extracted from? Okay, this is extracted from somatic cells, which are diploid, containing 2n, number of chromosomes. Less than 5% of your genes differ from others. Think about it. DNA is a nucleic acid composed of a double-stranded helix of nucleotides. There are only 4 bases in the human genome, A, T, C, and G. Thus, if it is not the monomers that differ between us, what is it? It is the sequence of monomers, bases A, T, C, and G. Each gamete contains 1 copy of each chromosome and thus contains 1 allele for each gene. After gametes recombine during fertilization, haploid chromosomes return to the diploid number and thus contain 2 alleles for each gene. Each individual's genotype contains 2 alleles. This isn't in your study guide, but I would assume you would know this already. Put this in order based on decreasing size: allele, chromosome, chromatid, DNA, gene. You start with the chromosome, then the chromatid, DNA, then gene, then allele. In pea plants, purple flowers are dominant to white flowers, and round seeds are dominant to wrinkled seeds. Which of the following crosses a plant containing purple flowers and wrinkled seeds with a plant containing white flowers and round seeds? So now we're looking at what parents there would be. Okay, so homozygous, heterozygous here, and homozygous, so capital P, little r is one parent, crossing with little p, capital R, little r. When describing the physical features expression of a trait, you're describing the phenotype. When describing the actual genes, alleles represented by letters, it is the genotype. What determines what an organism looks like? It's the genotype, which is found on the genes. What organism did Mendel study to determine genetic inheritance patterns? It was the pea plants. Mendel's law of dominance states that the recessive trait can only be expressed when an individual is homozygous for the recessive trait, as heterozygotes or hybrids always express the dominant trait. These individuals are also termed carriers. Why? They have a hidden allele and it wouldn't be expressed. Using the letters capital T and little t represent the dominant tall and recessive short gene in plants, write the genotypes and phenotypes for homozygous dominant, heterozygous, and homozygous recessive. Capital T is a genotype that's tall. Capital T, little t would be tall, according to the law of dominance, and little t would be short, according to the law of dominance. A red flower and a white flower cross to make pink flowers. What type of inheritance pattern is this? If the pink and white flower are crossed, what will be the possible genotypes and phenotypes of their offspring? Now, in this case, a little r is being represented for the W, being white. And of course, in other examples, we've seen it as W's. So here we go. So we'll get the red flower, okay, and the white flower, and we'll get incomplete dominance. 50% are going to be pink. If this is a pink flower, the intermediate, okay, and the white was recessive homozygous, and then 50% will be homozygous, which is white. So if you ever hear the word pink and snapdragons and some kind of an intermediate phenotype, something that's in between the dominant and the recessive, but only shows as that, that's considered incomplete dominance or intermediate inheritance. Now, cross a zebra that is heterozygous for thick stripes and a thin striped zebra. So let's see. Okay, heterozygous for thick, use capital T for thick, and homozygous for thin. Okay, so what percent of offspring are homozygous? 50%. Yes. Which percent of offspring are thinly striped? 50%. Yes, the phenotypes. What is the ratio of thick to thin stripes? 2 to 2. In certain cactus, prickly spines can be two-pronged or one-pronged. If a true-breeding one-pronged cactus is crossed with a true-breeding two-pronged cactus, the F1 generation displays both types of spines. Is this an example of codominance or incomplete dominance? Codominance, when they're expressing both of the dominant forms of the trait at the same time. And do a test cross to show the F2 generation. What's the phenotype ratio of the offspring? Okay. So let's see, we use T and O for true and one and two pronged. Okay, so they're both heterozygous. Okay, so we're going to get two pronged, capital T, zero, so one pronged, two heterozygous parents. Okay, so we get one to two, one two pronged to two one pronged, and one pronged spines. Okay. Distinguish between Mendel's law of segregation and the law of independent assortment. Mendel's law of segregation means that homologous pairs of chromosomes will separate and recombine to form offspring. In law of independent assortment, no control over separations, each separate alone. In radishes, the gene that controls color exhibits incomplete dominance. Capital R, pure breeding red radishes crossed with pure breeding white radishes make up the purple radishes. Again, purple's between red and white. It's the intermediate color between those two main colors. What are the genotypic and phenotypic ratios when you cross a purple radish with a white radish? Okay, so here we go. So you get 50% capital R, little r for genotypes, 50% little r. For phenotypes, it would be 50% purple and 50% white. Achondroplasia, which is dwarfism, is caused by a dominant gene. A woman and a man, both with dwarfism, reproduce. If homologous achondroplasia results in the death of the embryo, list the genotypes and phenotypes of all potential live birth offspring. Prove it using a Punnett square. Okay, capital A will be the dwarfism allele, and little a will be the normal allele. This is a dominant genetic disorder. Okay, so we'll cross those both, and we get half or 50% will be dwarfism, and quarter or 25% will be normal. And the ratio of dwarfism to normal is 2 to 1. So watch out when you have to do a ratio of phenotypes against what you're actually getting in the Punnett. Okay, so dwarfism to normal, okay, there's just a normal 1 to 3, kind of thing like that, but really this is here. Okay, so I think that would be all dwarfism. I apologize. I think I'm wrong here, right? 3/4 are going to be dwarfism. That's phenotype to 1 that is normal. I apologize. This should be 3 to 1. Okay, human balding is a sex-linked recessive trait. Illustrate the two possible crosses for a bald man and a non-balding woman in the Punnett squares and answer the questions below. Okay, so there we go. You can see that. We could have like a bald man, balding as a man who has a balding allele there. And then a woman who has normal, and that's the crosses that you get. And then you have a balding man, a man who has the characteristic of the balding allele on his X chromosome, and the woman who's heterozygous, and that's what you get. So let's say we know that the non-balding woman carries the balding gene. Okay, how many sons should be worried about going bald? 0 to 25%. What percent of carriers are possible? 50% to 75%. Why are more men bald than are women? Because not able to hide or carry the trait, because males only have one X chromosome. The Y doesn't have this gene on it. So if a woman knows that she carries a recessive allele, should she avoid bald men so that her potential daughters don't go bald? Why or why not? Well, if the woman is with a bald man, then there is a 25% chance that her daughter will develop baldness in some form. The gene for baldness is only found on the X chromosome, and again, not on the Y chromosome. So a male who carries a recessive form of this gene on the X chromosome will have no way to override the expression, and probably they will express this baldness eventually in his lifetime. Whereas a female who is heterozygous will, have it, but not have the actual expression of it, but will be considered a carrier to carry it on to her offspring. Now, a man who is IAI, which is A blood type for blood type, marries a woman who is IB homozygous B blood type. Provide the possible phenotypes, blood types of the offspring of this cross. What types of inheritance patterns does blood type exhibit? Okay, so here we go. This is the B blood type with the A blood type. And again, little I represents the O allele. This is a codominant situation. And like I just mentioned that, multiple alleles are codominants. Okay, the phenotypes are AB, B type only. Okay, yes, because we're getting AB, and B blood type. There's no chance of an O blood type there. And analyze the following pedigree for the disease of Budibiosis using capital T and little t. Write the genotypes of each individual. Is the trait autosomal or sex-linked, dominant or recessive? How do you know? Okay, autosomal for sure, and recessive because both parents don't have it, and not all the males have it. So it's autosomal. It's not sex-linked, and all the parents aren't having it. These parents do not have it. So it's recessive. Dad would have to be sick. Okay. So you have to backtrack, okay, and just see what the possibilities are. You possibly have to do a Punnett square to determine certain things, maybe working your way backwards. And that's like this one. Tammy has taken Danny to court for child support payments. Tammy claims Danny is the father of her 3-month-old son. Danny denies this. Judge orders blood testing done on all three. Tammy is heterozygous blood type A and her son is type O. Test across all possibilities, so you might have to make multiple Punnett squares. If Danny is the father, circle the possible genotypes he could have. Okay, so he's the father, and then the George blood test being done. Tammy she's IA. She's this all the way through, so just line her up. And then you look at all the possible situations. Put her son in the box. Put her son in one of the boxes and kind of see what you could be getting if you're going to get. Okay, so obviously, okay. So if the possible genotypes are IAI, because here's the boy, IBI, here's the boy, and II, here's the boy. So the father could be A, B, or O. Create a pedigree for the following X-linked trait. Johnny's normal, but he marries Carrie, who is a carrier for hemophilia. They have 3 children. The oldest is a girl, and the younger of the 2 boys has hemophilia. Label each person with the correct genotype, as you put it in the pedigree. If the second allele is unknown, just put a line representing that, because it could be, if it's homozygous, heterozygous, it doesn't matter there. Create a Punnett square to prove genotype of the oldest child of Johnny and Carrie. Okay, so those girls. She's a carrier. Okay, so she's there automatically. You shade her in halfway there as a carrier. Okay. He's normal, so he's all set up. So what's the possibilities here? So you set this up here on the side. Here's mom, here's dad. Normal, female. Carrier, female here. Okay, now you're going to get one colorblind. I'm sorry, yes. X-linked trait, if it's colorblind, could be any. But it's just here where this is going on here, where this person, one fourth or 25% could actually have the condition, show it as a phenotype, and this is a male that's normal. So these are situations where you're like, well, okay. So you can just leave them open like that. Okay, you use the pedigree below to answer the following questions. Okay, is this disease sex-linked? Yes, because the males are all showing the actual disease, and you see some carriers there. What percentage of this couple's possible daughters will have this disease? 0%. Carrier, 100%. Shade the 2 guarantee carriers that are not yet identified. Right here and here, because the boys have it. These have to be carriers. Okay, she was able to carry on this one to the female. Okay, so okay, these are her 3 kids. Yes, so she carried it over to her daughter here, and then the daughter actually carried that allele to her sons, and they have the disease. Okay, now if the couple with the son in the 4th generation, 1, 2, 3, 4. Okay, this guy here had another son. What's the probability that he will be affected with the disease like his brother? Probably 50%. Yes. How many generations are displayed in this pedigree? 1, 2, 3, 4. How many men have married into this family? 1, 2, 3. And how many diseased females, does this family contain? None. No diseased females because it's an X-linked trait. Okay, another name for a pure genotype like capital T is just homozygous dominant. This is just vocabulary review. Another name for a pure genotype like little t is homozygous recessive. So make sure you can identify those 2 when you see them in a question tomorrow. Another name for hybrid offspring is like capital T is heterozygous. That actual genetic makeup of an organism on its genes is called its genotype, and it's done by lettering. And the appearance of an organism, or what we see on the organism, is a trait like blue eyes, brown eyes, short hair, long hair, balding, not balding, is called its phenotype. So heterozygous round seeds, based on this information, capital R, little r, homozygous round seeds, big R, wrinkled seeds, little r, capital homozygous round seeds, capital R little heterozygous round seeds, capital R little heterozygous round seeds. Okay, an organism having 2 different alleles for a trait is called heterozygote. Mendel hypothesized that each trait is controlled by a factor now called a gene. The phenotype of an organism reflects all the traits that are actually expressed. If an individual possesses 2 recessive alleles for the same trait, the individual is said to be homozygous for the trait. Put the following terms in order from largest to smallest. Okay, so chromosome, okay, nucleus, DNA, and genes. Okay. A person who is heterozygous for type A marries a person who is homozygous for type O. What's the chance of them having a child with type O? 50%, because you've got AO, OO, and eventually there's OO in the box. Dihybrid crosses. Don't forget you're going to have one on the short answer tomorrow. So a cross that shows the possible offspring for two traits, like if you have fur color, capital B, black, white, capital R, little r, rough, smooth, coat texture. In this example, we're going to cross a heterozygous individual with a non-heterozygous individual, and their genotypes will be capital B, capital R, little r, crossing with capital B, capital R, little r. Now you need to do that tomorrow. You're going to hear it in this description, like here in this slide, and then you have to construct their genotypes like this. So find all possible gametes that can be made from each parent. Remember, each gamete must have one B and one R. So we do FOIL, BR, capital B, capital R, capital B, little capital R, and little b, little r. Don't forget to do that. Don't forget to do that. That's how they look when you put them on there. I'll walk around to make sure. I'm physically going to look around and see. If I see you going the wrong way, I'll try to point it out to you, and you'll have to fix it while you're sitting there. So what are the possible gametes for capital B, little r? Say it was that. That was the parent. So that would all be it. That would be the only way it would go, because there's nothing else that's different there to put in for gametes. What it would be for capital H, little h, capital T? Well, it could be these. Capital H, capital T, capital H, capital T again, then little h, capital T, and then little h, capital T. That's it. All right, so let's do this cross using FOIL. Arrange all the possible gametes for dad on the top of your Punnett square and mom down the side. These are a little off because of the slide. But I think we get it. Okay, that works out. I think that's fine. Okay, we'll figure it out. Right, that's good. Okay, and so we have that finished up. And then how many of the offspring would have a black rough coat? How many are going to be having a black smooth coat? How many offspring have a white rough coat? And how many offspring have, I can't read it. What does it say? Oh, yes, a white smooth coat. Okay, so there we go. Let's see. 1, 2, 3, 4, 5, 6, 7, 8, 9 for this first one. 1, 2, 3, 3 for this one. 1, 2, 3, 3 for this one, and 1 for that one. So it's 9 to 3 to 3 to 1 is the ratio out of 16. Then there's practicing some that you could do. This is just typical. There's another one that was just mentioned here about heterozygous for both traits, but then homozygous recessive for both traits. That's homozygous recessive for both traits. That's heterozygous for both traits. And then you see something like this. This is another way of doing it. Sometimes people like to do it with 2 Punnett squares, and that's a possibility instead of one big one that's 16. And just have this run through for you. So I'm going to keep going through this. Well, that's something you could do there. And so here comes another one. And this is doing that example with the 2 Punnett squares. And again, you can review this tonight if you think that'll help you. Just make sure tonight that you finish up everything in quarter 3, weeks 2 and 3 tonight, because that's pretty much going to help you to do the test. We haven't done a test in a while. Now it's been 3 weeks. So this is your first one that's worth 100 points. So it'll help doing everything with the test score tomorrow, and all that work is put behind you. Remember, this guy will go in at 60% of your grade. This will be it. The rest, everything else we've done in the first 3 weeks is just 40%. Now, be kind to me, Miss Fish, and do not have her make a special trip again to see your late working classroom. Just get it all in tonight. I'm keeping my eye on you. Course selection is soon, so the way you work and finish the work matters.